CCNTCSAGBHYK question mark. So welcome. This is cozy. We made this ball, and this is a 20-sided die. Sarah Hamilton and I put it together. We worked until 3 in the morning last night, so if I seem a little fuzzy, I'm not ordinarily like this. This has all 20 amino acids on it. We're basically working off of the quote-unquote genetic code. Many of you, if you're under 30, have learned this in high school, like in lower grades. Over 30, people are like, huh, I heard about it. And then there are only a few things that I want to say. First of all, I'm not sure what the real importance of the genetic code is as far as life. I mean, compared to, say, like the effect of sunlight, or how shallow you breathe, effect of friendship, compared to this one way of seeing life from the center of ourselves. But I do want to sort of like explore what it could mean, as opposed to what it does mean, as opposed to what it's claimed to mean. So we'll explore that. One of the ways we're exploring that is we're going to make our own sequence with this. Okay, this sort of rolls, and whatever comes up, that's a sequence. So first I want to teach you how to read what happens, because we're going to take turns rolling it and make a sequence together. I'm not going to try to mystify you. This is demystification, although it's remystification too in a different way. But let's just say this. If you have 4 base pairs, okay, the possibility for A, T, G, or C, right? That's that standard DNA thing. There's 4 base pairs, or like A goes to T and G goes to C. And that forms the letters of the DNA. Well, every 3 base pairs makes a triplet that becomes an amino acid. But there are only 20 amino acids. Along the lines of redundancy, many of these triplets, right, like CCT or CGA, you notice these are all alanine. These 4 here are all alanine. These 4 here are all glycine. These are valine, right? 4, 4, 4. Some of them, like, only have one, say methionine, AUG. There's only one methionine. And there's a reason for that. AUG, methionine, says start a gene. It's a start thing. So you can see on here, if you look on methionine, there's a crazy one. It's this M at the top with all the writing on it. M, methionine, ATG, or AUG if you're doing protein, the uracil replaces. Anyway, that methionine, when they search for genes, after making a big sequence of some organism, whether it's a human chromosome or a yeast cell or a worm butt gene they're looking for, they look for ATG or AUG, methionine, because that's a start. Because what we're going to do is we're going to roll a sequence, and what it turns into on paper, we're going to have a sort of drawing. We're all going to get some paper and pens. What we would do is we would probably, if we were doing a real genetic gamble and had an endless amount of time, we'd keep rolling this 20 or more times until we got to AUG or ATG. And then we're going to write down A, T, G, right? And then maybe in big, we'll write down we can either write down M, or we can capitalize the next letter. This is because I think we're designing a sentence. Not necessarily a sentence that makes any sense, but we're going to try to read it anyhow. not necessarily read it for use value, but read it as a sentence. So let's say if we roll this again, I'm doing a demo of what we're going to do together. Yeah? So I roll it again and I get an E. That's glutamic acid, right? Oh my gosh, I don't know where that Z came from. That makes no sense. The rest of it is fine. So what we'll be doing, and you'll be able to write this up any way you want, but you might want to write it in triplet form. Is
it the spelling?
this Z here?
Yeah, it's not on here.
Yeah, no, it just came out like on the computer, yeah. So anyway, there's two things. One, I would say this would then be a capital E because it's the first letter of a sentence, right? A gene being a sentence, maybe. We'll talk about that. And then over here, whoever rolls it, we're going to take turns rolling it, will have a choice between GAA and GAG. Like I said, glutamic acid has redundancy, right? There's 64 triplets that make up 20 amino acids. So some of them have 2. Valine here you can see has 4. Even serine has 6 different triplets. And you get to choose your triplet. Let me get back to where I was. I'm going to choose GAG, all right? Somebody want to roll it? You can only do so much on this table. We're going to go upstairs. But, okay. So, here's your test.
Why?
This is tough. What?
Why?
Y, okay. And it'd be, I guess, a lowercase y. Okay, and then which one of these tyrosines do you want? TAT or TAC?
That seems appropriate.
Okay, yes it does. We need one more roll. Can you roll in?
Yeah.
I think EY is not good. We're gonna be doing this together more. We're gonna try to make at least 30 sequences. Okay, so you got what is it?
C.
A C. Okay. And which one do you want? Which triplet?
TGC.
TGC. We keep rolling until we get to a stop. If you notice, we have the 20 amino acids here with the codons. And these, you can see they've substituted T for U, uracil for tyrosine, but that's okay, don't worry about it. A DNA strand, when it's sequenced, they do the same thing, right? You notice they have TAC, and you go over here, here's TAC, that's methionine. It also translates to AUG or ATG. Methionine says start. So what they're showing on this little graphic is every sequence starts with a start. And it goes on to valine, leucine, isoleucine, and then stop. Okay? You notice there's a stop codon here. Stop here, and stop here. And that's TAA, TAG, and TGA. Certainly what I'm not right about, definitely, along with this linguistic metaphor, is that since this can't be possible, is it? Since for stop codons, there's 3 possible stops to a gene, right? I figured that one is a period, one is a question mark, and one is an exclamation point, right? We've arrived at a capital letter E, Y, C, exclamation point, right? What are the different ways that we could figure out what that is, what that symbolizes, those letters, that sentence, right? Is there a Ike?
Ike, yeah. A tree.
A tree.
You write it differently, but you pronounce it sort of the same.
Sarah, I know you don't draw, but will you draw a tree? Okay. We're now drawing, yeah. There are various different ways to read a sequence, right? A standard way to read this would be, actually, if you find a gene and you isolate it and you sequence it, would be to actually try to knock it out of the organism that it's living in, right? But you first have to isolate the gene from an organism and then knock it out and then let the organism develop and see how its experience changes. The experience of its body forming, maybe its behavior in the long run. Another way that people study these sequences once they're sequenced is, and I think when my computer's hooked up I'll talk about how they're sequenced and how they're read. People look at something called homologs, which are samenesses. Searching for homologs, we would go to, and have it, there's a place called BLAST. It's an online software run by NCBI, and it's like a Entres. It's a open source to some extent, software online where you can, if you find a sequence, if you research a sequence, you can then type it in and see other organisms that have a similar sequence. If it's very similar, it's often considered to be an organism that's closer to us or closer to the organism you're studying in time of separation between species. So a lot of people will draw these maps, trees of evolutionary proximity of a certain gene and how it's changed over time. You know, because we have very similar genes as fish, but after we broke off from fish, the fish continued to evolve, and so did we, which includes like mutating. And so, how do I say, this is going to sound strange, but no organism on Earth is older evolutionarily than other organisms. Because we're all related to a similar organism, an organism that seems to be our ancestor from far back, like say bacteria, or probably we're protists, certainly worms. If we look at worms and we look at ourselves, we're worms with legs. The worms that we came from continued changing after we broke off from them. So the worm that you meet is just as old as you on the evolutionary tree. It's a strange thing to say. Now if right when we broke off, if there was a gene that we had and a gene that they had, they're the same gene, maybe only one thing mutated, like fingers, you know? Some early worm fingers. They have like worms that are showing signs of having early arms, right? Both genomes start to mutate, and they start to mutate often at, you know, a certain rate of standard, you know, hereditary mutation. Sometimes there's different rates. There's a rate of change, and then there's a rate of change, and there's a rate of the rate of change, and that sometimes changes really drastically. Is that called catastrophe theory? Right? Things, sometimes you have a wild new, like some people think that our brains got really big because we caught a swine flu that suddenly made us have giant frontal lobes and that we haven't stabilized, and that's why none of the birds really want to talk to us, because they're like, tell us when you're ready. This was one of my blasts that I did, and what we got down here was sort of amazing. I just typed in some sequence, and you can see over here, it says all sorts of different things, serpin peptidase inhibitor, etc. But over here in particular, this is bovine. D-A-N-R-E is Daniorario, which is to say zebrafish. And this is human. Right, so you can look, and this is a leukocyte elastase inhibitor. Okay? We're going to do a test. Would you like to draw your version of a leukocyte elastase inhibitor? It can be like right here. And you can do it in 30 seconds. It's not, this is not a test.
What's a leukocyte?
Witte bloedlichaam.
Oh, with look. Oh.
Is that for
Yes,
leukemia?
so.
Yeah.
Or-
Yeah.
And so it's immune, I think it's your white blood cells.
Yeah, that makes sense. So if it was an inhibitor of elastase, it would make you less elastic. The cell would be less like this and more rigid. And there's a picture of it. Okay. So what we're going to try to do actually together is make some art, making triplets together. We're going to roll this maybe 30 plus times. we're gonna come up with our sort of amino acids from that, and we're going to try to read the sentence with our minds and draw a picture of what the sentence means to us, each of us separately. We'll get separate pieces of paper. And then we're going to look it up on Blast for homologies, and we're going to choose 3 or 4, maybe we'll list 10, and people can choose either to draw all 10 or draw 3 or 4 of them, of what they think. You know, some of them might be more obvious. In other words, a putative, uncharacterized protein from a chicken. If this comes from a cow, we might have, say, like, would you draw a cow also, right here, and then an arrow to this, so that we have like the organism, and then the cow inside of it, or the organism that's living inside of a cow. Right? This is cool. Okay, so, you know, this would be, yeah, that's working. Right, so, and then it'll be optional whether you want to write leukocyte elastase inhibitor, but we'll get there. In any case, we're coming up with an iconography of a random text and an iconography of the actual organisms that this random text exists in already in the world of our knowledge. I think these make good pieces of art without having seen them. I think we're all going to make a good piece of art, and we can. I want you all to sign yours and consider either taking it home and hanging it on your own wall, or trading them with each other. Okay? Because I'm going to make one, and I want to trade. There are various ways to read DNA already, and I think we're going to come up with some today. But one of the things that I want to emphasize is there's a tendency towards utilization, and therefore most work in biology with genomics is based on getting results, you know, in one way or another. Not necessarily getting use value results. Most scientists are not actually utilitarian completely. They're actually game players that want to actually, they need advanced puzzles or they go crazy. But they tend to isolate useful bits that can be used over and over again. And how do I say, at the point in time when they find a useful bit through searching, right? Through a genome, basically sequencing one base pair at a time, using either radioactive or fluorescent tagging, we'll get into it. They make a sequence much like this, and then they test to see if it does something that they can verify. This is what you have to do in science. And then one of the ways that they test that is that they write down these letters and they send them in an email to a company that sequences the gene. We could send this sequence to an online company that does sequencing, and they will make it for us out of protein or nucleic acid, or even inside an infectious agent like a plasmid that could infect another bacteria. Our made-up gene could go into a bacteria. We may even try to do this. I contacted a company called GeneArt, a sequencing company. I like their name. And I said to them, hey, I'm thinking of rolling a sequence, you know, randomly. I don't know whether it's, you know, going to be even able to be sequenced, whether it can be stabilized inside of a plasmid or inside of another organism. And I don't know what it does because I'm making it up. We might be able to find some homologies, etc. But are you guys interested in producing a bacteria that has this random sequence in it? And they said, well, let's start slow. They said, you send us the sequence and we'll check it against our database for pathogenicity, which is to say deadliness, right? First things first. Let's see if you randomly roll something that we know is deadly, because they probably wouldn't sequence that, at least not unless we had a little more clout. The putting a gene in, which is transgenesis or transformation, is actually adding a gene, and then what you get is Generally, you get the result you're looking for if you have success, which is to say the cells turn green or the cells turn blue, or the milk cells produce more milk. And then you would re-inject these cells, maybe into a growing embryo or an egg, or re-inject them into a living organism. Strangely, there is no center. In other words, when you're an embryo, if you cut an arm off and put it in tissue culture just like that, not a tissue culture, but a explant culture, the arm continues to make itself more of an arm, even without the rest of the body or the head. So there's no center. And I have a question as to whether or not, looking at a cell from above, it looks like an egg with this nucleus in the center, that we found the DNA in the center, and we found that it seems to be having an effect. We gave it this code of life rhetoric, but we're just dealing with basically egg yolk. And maybe inside the egg yolk is a lot of stuff that's utilized by the cell. It doesn't mean that it has a central role in commanding the cell. Sounds strange. There may be organelles that are doing a lot of interesting things. The Golgi apparatus is like a social for proteins, right? They're hanging out in there and going like, hey, you want to fold like this with me? You know, that kind of thing. There's a lot more going on. There's something called alternative editing. So a single gene can be edited alternate ways. We're already getting to, I don't know, in the media trade, what is that called? Hypertext, right?
Transmedia.
Transmedia. So already the genome is transmedia, okay? It's not like every sentence is completely copied out the same way each time. There's some wild stuff going on in some genomes, like where it can be read in all 6 reading frames and things like this. Nobody knows how it happened. It goes back and forth on the same gene in sequences like, it's crazy. Effects of genes and the proteins that they make are multifactorial, which means one protein doesn't just do one thing, right? There's multiple proteins, but what does your body do with the protein? Where does it fit in the puzzle of making you move around? Different people have different responses to different proteins, which is to say, you know, one person might have a serious allergic reaction to certain proteins in peanuts, right? Or fish. You have, say you have 30,000 genes, and some of them have alternative editing strategies built into them, so they go to different places or make different proteins from their transcriptions. But how do I say this? Those genes don't just come on at a steady level. Sometimes you make a lot of them, and sometimes you make less of them. And you know this. You know this from when you get drunk, you know? And suddenly, like, your body goes into, like, I have to process all this alcohol, boy, I feel drunk, oh, I have to run and pee. And you have to get rid of something that's been processed. What happens is, if you drink alcohol, genes that say process alcohol know the alcohol enters your body. They sense it. Something sinks into your genome, not in every cell, maybe in your stomach lining cells, and says, hey liver, get ready, we gotta process some alcohol, right? And they send some proteins out and the liver starts producing alcohol digestins. The symbol is red, green, and yellow. If the gene expression stays the same, it's yellow. If it's green, it means there's more. If it's red, it means there's less. And this is over time. They'll take a blood sample from a human every 10 minutes. And during those 10 minutes, anything is science. And in other words, they take a blood sample from you and they run a chip of the genes, and that's the control. You just come in, assuming that you're in control when you just come in, you know, like a blank slate. And then they can whip you with wet towels and tickle you for 10 minutes and take another blood sample. And you'll see reds and greens and yellows, all kinds of changes. 30,000 different changes of a little more, a little less, a wavering. It's almost like a music, you know, like some crazy jazz is going on from being tickled. There's a lot more going on than a simple sort of like one-to-one, but also a simple, we can attribute all this to genes, because things are happening in the world. Our genes do respond to them, and the world responds to our byproducts too, 90% of our personality, maybe 99% of our personality is genetically determined. So that means everything you do and everything you say, everything you think and everything you feel is actually pre-programmed by your genes to allow for that. It's vastly depersonalizing. Our bodies are built to sit in chairs at this point. And it's not because of our genes, it's because our culture says we reward people that can sit longer, right? If you can sit for 12 hours a day instead of 6, you're gonna go up the ladder higher. And literally you can see it in our bodies. I have it too, like a little slouch. And basically, I'm talking about an alternative way of reading DNA, and I'm talking about some of the complexities involved in the idea of DNA as being not just a language, but a master molecule, right, that masters us. What it means to be an organism, a whole organism, is still a question. Like a lot of biologists study whole organism and try to figure out what that means. And that's a lot more complicated than just the genome. Literally, I want you to think about the difference as we roll this between the idea of random chance and the idea of a gene that works, a gene that does something. And this is going to sound strange, but if you look into Darwin, one of the most frightening things about the theory of evolution is that it's spontaneous, random, arbitrary, and just based on chance. That every form of every organism on Earth, supposedly, I mean there are other ways of reading this, of course, but is based on random mutation and then the test of the environment. Whether or not the environment is that harsh, I'm not sure. I think we have actually a less harsh environment than we imagine. And that's why there's 40,000 kinds of butterflies instead of just one that survive the Earth. They have different niches, etc., but they have time to say I want to go really multicolored, lots of different colors. There's room for sexual selection, right? There's bird songs. There's no reason, as far as utilitarianism goes. People try to find reasons, but there's no reason why life should be so diverse if it's really so competitive. Actually, there's a lot of room. It might be that we just like to compare it to our system of scarcity. There's less of it here than where I come from, where becoming homeless is actually a pastime for our population. It's the idea that there's not enough doesn't necessarily carry over into the life world. I'm just putting this out there. There might be places where there's a lot of plenty and leisure. Plenty and leisure for a sloth, plenty and leisure for a caterpillar. Sounds crazy, but there's a lot of leaves to eat. And yeah, sometimes the bird pecks you to death, but until then you were just chilling on a leaf, chewing and living, and sort of playing around, getting mutated on some weird leaf. But I think they invent the flus these days and the vaccine, and it's your choice which one you're going to get. Are you going to get genetically modified to get the flu or genetically modified by the vaccine? And the government's sort of the world government is well, either way, you're going to become one of 2 mutants that we have in mind for the human race. And I'm not sure which one's better. I assume that they're saying the people that are vaccinated, A, will survive the plague or something, or B, will have some kind of resistance that makes them into what? Super conformists or superhuman white collar uptight people. I'm sort of what about white people, working class people? The enigma of chance opens us up to jazz and improv and spontaneity and poetry, which is part of art, and we're going to make some. But it also opens us up to, it's going to sound strange, to the void of the unknown, right? The true place of enigma. It may be very wide. scientists and artists like to go there. They stand on either side of it and look into it with different eyes. But they look in the same void. And along that line, I'm not very much a spiritualist. I don't know if you noticed. I'm not a materialist either. I don't think we're just material, and I don't believe in a soul. So I'm really confused. Okay? What the fuck is this? I don't know, all right? But look, along with art and enigma and spontaneity and jazz comes speaking in tongues. I don't know what it's called here in Dutch. Glossolalia. When people like Pentecostals down in Texas go, do your hyper-Orthodox Puritans do that here? No speaking in tongues? You know that? When you get the spirit?
Yeah.
But also, I'm hoping you will be able to access, through this process, a reading of these. And at the same time, we're going to be using blast homologs to find some of the actual places that a random sequence exists already in life. That's weird, isn't it? We're going to roll one randomly from this, and we're going to find that it exists in different organisms to a lesser or greater degree, you know, for more different kinds of genes.